So welcome everybody to the Funk Lecture Series on Biogeography. We're just going to wait a few minutes while everybody connects. I'm Karen Fowler from the International Biogeography Society, and joining us today are Oskar Hagen, Rohit Chakravarty, and Sandra Noguez, and Seda Eckert, who is behind the scenes. Nina Witteveen is not able to join us today, but she's also an award winner. And while we wait, I'm just going to go over upcoming events. Our biggest one coming up in the near future is the 10th Biennial Conference in Vancouver, Canada, which is happening in January 8th to 12th, which is very exciting. It'll be our first, well, it's a hybrid conference, so there are virtual options to attend as well as in person. And it'll be the first in-person one for us for a number of years, which I'm super excited about. Also, Vancouver's pretty. If you are interested in attending, there are late abstract submission is still open, but for in-person posters only, and virtual and in-person workshop sign up is still open. And for this session, this is how the Q&A is going to look. Each speaker will give their presentation, which is about a 10 to 15 minute talk, which won an award at the Early Career Biogeographers Conference, which was a virtual conference that the Society held in October. It was in October. I was thinking September, but they blur. And how we'll do questions, there'll be talk, and then we'll do a Q&A for each talk afterwards. You can put general comments at the bottom of your screen in the chat box if you want to say hi or just congratulate. If you want to ask a question personally, raise your little hand icon and we will promote you to a panelist and you can ask the speaker the question directly, face to face. And then if you want for us to ask your question for you, you can type your question in the Q&A box. And you can write your questions at any time during the talk, but we'll only ask it during the Q&A session. And with that, I will turn things over to Sandra. Sandra.
Hello, everybody. First of all, thank you, Karen. And first of all, congratulations to the 3 winners of the Early Career Conference. And our first speaker is Dr. Oskar Hagen, that he currently is a postdoc at the German Center for Integrative Biodiversity Research. And Oskar is a biologist, a computer modeler, environmental scientist, and he's very interested in investigating the emergence of biodiversity across scales of time and space. And today he will be giving us the talk that gives him the first prize in the conference. So all for you, Oskar.
Thanks a lot, Sandra. Also, thanks everybody for joining. Cool. So today we'll talk about, let's see if this is working, about deep time dynamics, eco-evolutionary processes, and the uneven large-scale biodiversity patterns across tropical moist forest. And my name is Oskar, and I will mostly talk about 2 recently published studies. One tackling a large-scale biodiversity pattern, and another one describing a little bit more of this novel methodology developed to better understand these patterns. And I first will present the general motivation behind my work, including my research question, as well as objectives. And then I will explain a little bit about the biodiversity model I developed to tackle these questions. And then I will focus on a specific biodiversity simulations, mainly looking at the pantropical diversity disparity. And I will finish it off by concluding and discussing some limitations and perspectives. So we know that biodiversity is unevenly distributed around the globe. As you can see in this richness map of vascular plants, with some places with few species and other places with many species. And this diversity pattern has often very characteristic patterns, such as the uneven biodiversity across tropical regions, which we call the pantropical diversity disparity. And you can notice, for example, that Africa has a much lower richness than the Neotropics or the Indo-Malayan tropics. And this led Richards in 1973 to call the Afrotropics even the odd one out. And I'm interested in general on why are there such distinct patterns, and also what processes shape biodiversity. So tropical moist forests are the most rich species terrestrial biome on the planet. And this low Afrotropic richness compared to the Neotropics and the Indo-Malayan tropics are shown here, for example, for groups of palms, but this has been shown to consistently hold across different other groups. And there are many different suggested hypotheses to explain this pattern, such as differences between current climatic conditions or past environmental conditions, or the contrast either between a lower speciation or a higher extinction in the Afrotropics compared to these other two tropical regions. Well, we looked at empirical data, so we sampled or we collected data for plants and vertebrates in order to investigate this tropical disparity. And you can see here clades of plants and how they're dispersed across like the Indo-Malayan, the Neotropics, or the Afrotropics. And you can see here, for example, each of these points would be a single group of plants. And here, for example, we're looking at palms and we have the richness. So you see, you have a higher richness in the Indo-Malayan compared to the Neotropics. So this point here is more towards this blue triangle. And this is just a way to see how these groups that are occurring in all of these three regions are distributed. And we found out that this pattern holds for 72% and 81% of the vertebrates. And looking at current climatic conditions, we found weak support for a general relationship between richness and climate while using a generalized least squares models accounting for spatial autocorrelation. The problem of looking at this through these correlative approaches can be tackled or can be investigated from the other hand, while we perceive or we look at biodiversity as knowing that these biodiversity patterns, they emerge from multiple processes, such as the interactions with biotic and abiotic conditions, speciation and extinction events, as well as adaptations to ever-changing past environmental conditions that such as continental drifts, mountain uplifts, and changes in temperature and aridity. And we know that all of these processes, they interact in complex ways that have to be considered in order for us to move beyond statistical approaches. And this complexity is a strong limitation to our mechanistic understanding. But luckily, we can have computers and we can build computer models to assist the answering of such questions, since experiments at deep time and at global large scales are hard, if not impossible, to reproduce. So to assist the answering of this question, I developed Genesis, which stands for a general engine for eco-evolutionary simulations. And it is a framework that allows eco-evolutionary feedbacks at different time scales while considering these dynamic landscapes. And it's implemented in R and C++, striking balance between speed and code simplicity. And the idea of this framework is basically to allow scientists or researchers to formalize distinct biodiversity theories, create and test this hypothesis, and allowing these historic simulations and future projections, helping us to quantify these processes that come from different disciplines and shape biodiversity, and thus enabling us to investigate spatial and temporal biodiversity processes. So now I'll jump basically on how the model works, and I'll give a quick overview. So the model consists of mainly three parts. First, we have inputs, which are dynamic landscapes, and a configuration file containing the initial conditions and the eco-evolutionary rules. And these are then fed into the engine, which computes and records species abundances, distributions, population divergences, phylogenies, and trait values, providing several different outputs that can be compared and contrasted to empirical data. So now I will show one example of such a landscape that we use. So here you can see the Earth back on 200 million years ago, and you see here air surface, temperature maps, and aridity maps, and these are computed and reconstructed using plate tectonic movements and topographical changes, lithological information, stable isotopes, and fossil data. These dynamic landscapes are represented here by a more simple landscape, which contains basically some highlands, a lowland, and a river. And to show how the model works, I will just use a single brown fox population. So here I'm talking about one single species, but you can imagine that we are accounting for the buildup of different species emerging throughout the simulation. So basically we start here at the speciation module where connected populations exchange genes, and these connected populations gain a genetic divergence. And if these populations are isolated for a divergence threshold or a certain time, they will speciate. But if they get into secondary contact, their genetic divergence will decrease. Okay, so in this case here, in this example, there is no speciation event. Then this species will disperse in the landscape based on their dispersal ability and landscape characteristics, such as barriers, for example, here, the river. And after dispersing, this species will suffer trait evolution. So here you can see the appearance of red fox and white fox variant. And entering the ecological module, these abundances are calculated due to biotic and abiotic interactions with possible local extinctions. Important to highlight here is that all of these modules are modifiable. So a user can basically change the rules on how species or populations of the same species gain genetic difference. So for example, it could be dependent on temperature or certain species traits. Dispersal can be stochastic or deterministic, or the landscape could offer different resistances, or trait evolutions, whichever traits one decides to model here, remembering that traits have to be linked with all of these other processes, or ideally would be linked to all of these other processes. So for example, you can allow for dispersal ability to evolve, while dispersal ability will then change how speciation process happens and how species would occupy or move around this landscape. Well, here is one example of one simulation that we run to tackle this PDD pattern, showing species richness emerging over time. So we start here with one ancestor at 110 million years ago, exploring different niche width, dispersal abilities, and divergence times with an evolving niche optima and excluding arid sites. So you see here through time the accumulation of richness. So this pattern, this X here, this bar shows richness patterns, and you see the emergence of these biodiversity patterns. So going back now to the empirical patterns, you can see after simulations are run, one single simulation here is one point in this space of PDD pattern. So here I show one example of such a simulation and how this richness pattern looks like. But important here is not only comparing one single emergent pattern of biodiversity by looking, for example, at palms and trogons, but looking at multiple emerging patterns. So what we did here is also we looked at the evolutionary history and considered net relatedness index. So it's basically a way to see how communities are nested within. So we consider a phylogenetic history. One example here is, imagine a community composed by these two species. They will have a high nestedness while a community composed by these two species would have a low nestedness. And here you see this spatial map of this nestedness components, and here a best matching simulation. And you basically see also here one example of a simulated phylogeny and the reconstructed phylogenies for these specific groups. So after doing all of these matches and exploring this huge range of parameters and simulations, we could show that past climatic variations are crucial in explaining this pantropical diversity disparity. And basically, by looking at these mechanistic simulations, we have hints that this Afrotropical lower diversity is driven by both a lower speciation and a higher extinction rate compared to these other regions. And this is probably related to the fact that the Afrotropics was exposed to a more extreme cooling and aridification, which with a much less historical environmental heterogeneity. And you can imagine this, or this could be also probably linked with the fact that the African continent lays on top of a tectonic plate. And one way of mechanistic exploring this, and the next step was, okay, so if we find these settings and these conditions that explain this, we go back and we change the input landscape. So here in one example, we erase the Andes. So we remove the topography from South America, and we run simulations again, and you see by removing this heterogeneity, or in this case, just the temperature gradient caused by this topography, you see that you reduce the neotropical diversity of tropical moist forests. On the other hand, when we remove connectivity between island systems and the Asiatic continent, or even removing heterogeneity, you also reduce diversity of the Himalayan tropics, while removing this aridity constraint from the Afrotropics at different time steps, we increase the richness of the African Afrotropics. So reaching out our conclusion, we see that deep time environmental dynamics coupled with eco-evolutionary rules, specifically a niche conservatism, can explain this pantropical diversity disparity, but it's still, we have still to investigate and look at different paleoenvironmental reconstructions, biodiversity data, and even model complexity and computational times, with our still strong limitations to the full exploration of deep or past environmental dynamics. But these tools are very promising. So using these mechanistic eco-evolutionary models and paleoreconstructions provide a way to catalyze interdisciplinary research and hopefully advance our numerical knowledge on biodiversity. And with that, I would like to thank all the collaborators involved in this research and people working at different regions and at different stages of this research, the landscape ecology group in Zurich and so on. I think I will be a little bit short because I think I'm approaching the time, and specifically thanks all of you for tuning in, and I'm looking forward to interesting questions.
Fantastic, thank you, Oskar. So time for questions. The first question.
Okay, Richard's 1973 paper is of course super famous. So have all his suggestions in terms of the explanation, everything that Richard said, which is, you know, Pleistocene effects and perhaps also Miocene cooling, but he mostly focused on the Pleistocene aridity, on climate changes. It all fits with your findings, or is there anything, any aspect in which he was wrong?
Well, I think that's a very specific question, and I think I can already say beforehand that probably it's also a question of resolution. So I feel like most of these is supportive, and especially about the cooling effects and so on. So here, I think the main findings is really to show that deep time has a strong impact on it. But you would have to be a little bit more specific about, like, I cannot answer if all of them are holding or not. But
my question is a very broad one. You kept mentioning the mechanistic understanding which you aim for. Okay, and so my question or my feeling is that this is a historic science. Would you agree?
Yes.
Okay, so how can there be a mechanistic understanding that goes beyond studying climate and tectonic movements, which you have done, and you do it better than Richard ever could. Okay, but how can we progress in having towards a mechanistic understanding? I just don't get it.
That's a great question. Thanks. I think the idea here is basically we don't start with the biodiversity patterns that we observe now, but we basically say we start with the best that we have, or the best that we know, how these environmental conditions are and how these species were distributed, and we add like the best of our knowledge, or we test these rules, right? So the idea is we then simulate forward in time, and we know which mechanisms are there, right, to explain these patterns. But of course, each single realization of this is an n=1. So I mean,
yes,
what is happening on our planet and what we observe here is one realization of this multiple range of patterns. And I think the mechanistic way derives from the fact that these simulations are not statistical or correlational simulations, but we are adding the knowledge, or at least the best of the knowledge that we have now, and the best of the knowledge of these reconstructions, and how the species interacted with these changing conditions. So, and I think, of course, there are many ways to answer this, and this is just one way of putting together hypotheses and theories, but I think many of these cannot be disentangled, right? And I think progress can only be made if we start then looking at more patterns, at different signatures of diversity, and we can validate certain mechanisms through, for example, genetic data, historical occurrences.
Thank you.
Thank you both. Yeah, let's keep going. We have two more questions, Oskar, for you.
Cool.
The next one is from Jorge Noriega, and he says, "Hi, Oskar, nice talk. Do you think that these biodiversity and spatial patterns could be different if you use data from insects?"
That's a cool question. I mean, I haven't looked at the data from insects, so if they hold, I don't know. I have no idea. I would say that a lot of the simulations, when we look at the groups of organisms and the parameters that we explore, these are things that make sense, at least for this range of organisms. So dispersal ability has a certain range, divergence time until speciations has a different time. So probably, and I think then looking at these different aspects and looking at more precise groups would be interesting. Yeah.
Excellent. I'm just worried about time. Okay. And the last one that we have here in the question and answer box is anonymous and says, "Has this been used in practical applications for land resource managers to date?"
No. No. And I think it also goes, I mean, I feel I would like to talk with Susan longer, like I felt I couldn't answer her questions exactly, but I think this question goes also in the same direction, because I feel we are still very far from understanding really what would be the implications, but I do think that such models help us putting together all of these different theories, this evolutionary thinking, this ecological thinking, and how these processes interact with the changing environment in order to better understand, for example, how climate change or fragmentation might impact biodiversity. So in this sense, I think there are many possible uses, but I see this still as a tool for inviting people to test different hypotheses and theories and to further explore. So hopefully, yes, and I think that would be a long-term goal, and it would be amazing if such tools could be at some point be used to predict biodiversity loss or biodiversity responses to changing climate, considering how the species interacted and reacted to past conditions, right?
Okay. There's no more questions and answers. Suzanne put here the reference, so for you to copy later. And then before we move on to the next one, I see some people that raise the hand. Okay, so Florian, yes, I think that you wanna.
Yeah. Yes. Sorry. Can you hear me?
All good.
Okay, great. Thanks very much for the presentation, and very interesting and thought-provoking again. So I'm very personally interested also because I worked during my PhD thesis on the project of trying to integrate dispersal ability in species distribution models, so bringing mechanistic approaches for this, accounting for dispersal capacities, and climatic more statistical models. And so I have two specific questions, really general, just by interest, but because I had quite a hard time to develop that on the specific model species I was looking at, which is bryophytes. So it's a bit similar to the previous question about insects. What do you think about because you were talking about the resolution that you were working at, I guess, given the large scale of the study, it should be like on the arc minute level, or I don't know to which level this is precise, and what do you think about the outcome that could or the perspective that you could have using the same the framework, but looking at more, let's say, microecologically or microhabitat based model species.
Mm-hmm. And can I answer this question? I think I get one question from this. So you are completely right, and about the impact of resolution. So this was always like a big trade-off, right? Because if we want to understand how things evolved and how things interacted in such a deep time, we are at the end very restricted to the resolution and the quality and the amount of data we had from the past. So we had to do some sacrifices there about the resolution. So it wouldn't be fair to assume we have a super high resolution and we know how environmental conditions changed in the past, doing basically an interpolation. So here was a little bit of a trade-off. And when we did this, we have to think that this dispersal ability or these events of a dispersal are not only, I don't know, in the sense that you were talking, but most of the time a dispersal would be a very individual process, or even within a shorter time step. And then you have like climatic seasonal changes and so on and so forth. So yes, resolution should change, and this relates more or less with what I'm working right now. So I'm trying to use the same framework and of course tuning evolutionary parameters and dispersal parameter to a much smaller resolution. So we did some work as well, for example, trying to look at the reindeer, the caribou distribution in North America. So within one species at which a much shorter time scale, and we have a much better resolution of climatic conditions. So we know, for example, how corridors were formed, and we could predict very well, basically, the genetic distance between these current populations based on these climatic conditions. And there is definitely an application, but most of the time we are really limited by data. So yeah.
Okay, thank you very much for your answer.
Thank you.
No, it's great. I'm very interested. I think that's really an answer that's opening many doors, and I will certainly have a deeper look soon and see how I can maybe try to put on or to answer some of the actual project I'm working on right now. I think it's connected very much. So thank you again very much.
Feel free to contact me. Yeah.
Oskar, very quickly, we have another question that is very much linked with that, I would say, from João Paulo dos Santos Vieira. And he's asking, hi, Oskar, thank you for your presentation. Have you run this model for finer scales? Do you think that there is any scale limitation for the algorithm you built? And needs to be short, the answer, because we need to move on for Rohit.
So I tested different spatial and temporal resolutions. Most of the time, you have to think which broad scale. So if you want to run a simulation for 110 million years ago at 0.01 degree resolution for the entire world, forget it. There is no memory, there is no computational and data. But we were improving in temporal, spatial resolution, reconstructions, computational time. So theoretically there is no limitation, but you have to imagine the practical limitations would be computational time and data, right? I mean, but you can always use theoretical landscapes and build it up there. But yeah.
Brilliant. So thank you so much for your talk and answering all the questions. And now it's time to move on to our second speaker today. Hello, Rohit. Remember to
Hi,
mute.
Sandra.
Brilliant. Hi. So my pleasure as well to introduce Rohit Chakravarty, that he's currently a PhD student at the Leibniz Institute for Zoo and Wildlife Research. And Rohit's main interest is understanding the natural history of Indian bats. And basically his ultimate research interest falls mainly in the categories of movement ecology and biogeography. And as well, Rohit was one of our winners for the last early career conference. And all for you, Rohit.
Thank you, Sandra. Welcome to all of you. Thanks for attending this talk. And I have a tough job to carry on from Oskar, who presented a really good talk and which covers a lot of ground in biogeography. I'll be a little more specific, and I'm rather new to the field of biogeography, but I hope you'll find this interesting. It's a presentation it's a study that comes from my ongoing PhD thesis. It's the first chapter of my PhD thesis, and which deals with the diversity of bat communities in the Himalayas. So as biogeographers, and if there are no biogeographers in the audience, and or there are those of you who are not biogeographers, but are ecologists, you would have read this cliched phrase over and over again that elevational gradients are excellent natural laboratories, just like islands, because at short distances along elevational gradients, you see a massive change in climatic conditions and vegetation, and the animals, the fauna obviously responds to these changes, and they organize themselves, their distributions along these elevational gradients. So it's a very good place to answer questions about why are species where they are, which is a very fundamental question in ecology and evolution. And in the literature, you would find plenty of cases where species richness is described across elevations and how it varies across elevations, but the same cannot be said for functional and phylogenetic diversity. And functional diversity deals with the traits of different animals, and phylogenetic diversity deals with their evolutionary histories. When these two are combined, these two elements of diversity are combined, they give insights into how these communities established themselves post-colonization, and they also allow us to predict the future of how these animal communities will respond to anthropogenic changes, and particularly climate change. Believe it or not, the Himalayas, which are the mightiest of mountains and the largest elevational gradients, are still not quite thoroughly explored with respect to the patterns of diversity across elevational gradients, and more specifically for elusive taxa such as bats, which are globally present a research gap. Now, why am I studying bats? Is it just because I'm interested in bats and I want to throw a taxonomic perspective? Yes, correct. But there are other benefits to studying bats. For example, they are the second largest order of mammals in terms of species. They're the only mammals that can fly, which means that they can shift their distributions quickly in response to climate change, and that can lead to altered community structures. In previous studies from Africa and from South America, we've not got consistent results on how bats are distributed along elevation gradients. So there's a need to study more mountains to find generalizable patterns, if there are any. So we asked how does elevation affect the taxonomic, functional, phylogenetic, genetic diversity of bats in the Himalayas. There are two main expectations that come out of theory, and I'll briefly run you through that. Now, in the first case, we're looking at harsher environmental conditions. And in harsh environmental conditions, if you imagine every dot in this figure to be a species, represented by certain traits given by the x and the y axis, you would expect that harsh conditions homogenize the traits because certain traits are required for surviving in these conditions. And oftentimes these traits are also linked to phylogeny, they're correlated with phylogeny. So what you end up is a sort of filtering, a sort of homogenization based on both the functional traits and the phylogenetic traits. And this is called the environmental filtering mechanism. You would expect this at higher elevations. Whereas in the lower elevations, where the conditions are more benign and there are more species, you would expect interspecific competition to drive some of these patterns, to drive trait differences. And interspecific competition typically diversifies traits and it also leads to extinction of or removal of, phylogenetically similar species. So you end up with what is called functional and phylogenetic overdispersion. And this is an outcome of the limiting similarity mechanism, which you would expect in lower elevations, like I said. And of course, there are other patterns, that there are also other patterns that could come out of these. I'm just presenting, for want of time, I'm just presenting the most probable right now. And we studied these bat communities in Kedarnath Wildlife Sanctuary, which is in the western part of the Himalayas. And for the last 5 summers, I've been doing bat surveys here. We've studied bats from 1,500 meters to 3,500 meters by netting them to get their trait data. But also, we put out bat detectors to record their calls and identify them. And as you can imagine, this is useful, particularly in areas like these, which are treacherous for bat trapping. But with the help of recorders, you can get more data on their occurrence and their abundance, but with caveats, which I'll discuss now. Our trait data comprised 4 morphological traits, which we obtained by catching them. We took standardized photos of their wings, and the wing parameters and the echolocation call parameters give a good indication of how bats forage, which kind of habitats they live in, etc. But now, like I said, trapping bats is harder at the high elevation, so we have to depend on acoustic data for the occurrence, which means that not all species can be identified to the species level from calls alone. So we had to lump functionally similar species into guilds, or sonotypes as we call them in bat biology. So I would argue that this presents a conservative estimate of functional diversity. It affects the magnitude, but not necessarily the pattern itself. Similarly, for phylogenetic data, we use the data from a published phylogeny because some of the species from our study area were not represented. We had to replace them with the nearest sister species. And because we're looking at sonotypes now, we had to represent one species for each sonotype in the phylogenetic data set. So again, conservative, affects the magnitude, but may not necessarily affect the pattern across elevations. And then we went on to measure the 3 different elements of biodiversity: the taxonomic diversity, functional and phylogenetic diversity. The data I'm going to present are the ones that are underlined. So typically with respect to functional and phylogenetic richness and dispersion, and I'll explain those. Then because functional and phylogenetic diversity are also highly correlated to species richness, you need to do null models to understand whether the observed is different from the expected. I'm not going into too much detail, but this is typically in community ecology given by the standardized effect size, which is given by this equation. And you'll understand it when we see the plot. So first thing, simple result. We're dealing with species richness now, the taxonomic diversity, and as expected, it declines with elevation, which is also a finding from other mountain systems in the world with respect to bat. And to help you understand the locations, there's a small schematic diagram here, so you can refer to that to understand the locations and at what elevation they are. Now, when we get into functional diversity, the patterns start becoming a little more interesting. And over here, you have the SES values, the standardized effect sizes with the location. With respect to our data set, you interpret it in this manner, that an SES greater than 1 indicates over dispersion. So there's so the traits and the phylogenies that are more dispersed are higher than expected. An SES value less than -1 indicates under dispersion, and when it's between 0 to 1, it matches what is predicted by the null model. So a pattern that immediately stands out is that at the higher elevation, and again, another thing to point out, is that the highest elevation is excluded from this data because we only had 2 spatial replicates, which were not sufficient to perform null models. So the highest elevation now is at between 2,700 to 3,000 meters. The pattern that stands out is that at Chopta, all the functional diversity indices are less than are under dispersed, and this indicates that they're likely filtered by the environment. Certain traits are likely filtered by the environment, whereas in Mandal, where we expected some sort of over dispersion, you see that the functional richness is quite high, but the dispersion is not so much. And which means that if you look at it, imagine a trait space, the traits are sort of packed, and this is called a niche packing mechanism. And to put it simply, there are different kinds of traits in the bats in Mandal, but they're not necessarily very different from each other. Now, to prod this further, we redid this analysis, but by removing a family of bats called the horseshoe bat. Now, the reasons were twofold. A, these bats have very different traits. Their calls are extremely different. Their call structures are pretty different from the rest of the bats in our community, and they're only found in the lower elevations. So after we removed them and we did this analysis, we saw that the patterns shift quite a bit, which indicates that the rhinolophids or the horseshoe bats have they sort of pull the trait space in a certain direction. Now, this makes even more sense when we look at phylogenetic diversity and see that there's absolutely no change, whether we're looking at all the species, and even after we're removing the rhinolophids from the data set, we see that there's no real pattern of phylogenetic diversity with elevation. And this is because the horseshoe bats belong to only one family. In fact, there are 4 species in our data which belong to the same genus, but they have very significantly different traits from each other, as well as from the rest of the species in the assemblage. So when you remove them and redo the phylogenetic diversity, you don't see any pattern because it's only the loss of one family, but the traits are a lot of different kinds of traits are getting lost in that. So, and that's perhaps the reason why you see such level of under dispersion in Chukta. So to conclude, we observed that environmental filtering is likely plays a role in structuring the higher elevation communities. But at the lower elevation, where we were expected a lot more competition, we observed indications of competition. We observe a niche packing mechanism, and these patterns that we see in the data are dominated by the horseshoe bats, which are closely related species but with extreme trait values. And I'd like to end by thanking my field team, my scholarship, and a lot of funding organizations that funded this work. And this work was published just 3 days ago, so you can read and provide more feedback, ask more questions, look at the limitations, most important. And now the ball is on your court. So I'm happy to take questions.
Thank you so much for the talk, Rohit. Okay, so we have already one question from Trevor Price. He says, what happens from 300 meters to 1,500 meters, for example, at Dehradun?
Okay. Yeah, that's a good question, Trevor. And I wish my elevational gradient, the valley went even deeper. It would have helped understand some patterns a lot better, but it's very hard to find an elevational gradient going right from the foothills to the tip of the mountains, a single contiguous elevational gradient in the Himalayas. I would expect, having done bat surveys around the Dehradun, there are a lot more species, and but I haven't looked at their traits, but it seems like the phylogenetic diversity is definitely the same as at 1,500 meters in Mandal. The functional diversity as well, if I remember the species correctly, is not very different from what we've seen at 1,500 meters. In fact, I would say it probably does not affect the patterns a lot. The patterns are not very different further down in the elevation.
Mm-hmm. Before Jorge, Trevor put another one, and I think that is linked with what you were saying, and says three dots, and said richness is highest at the bottom.
Yes, richness, because these were surveys I did in 2016, and they were done rather in an unstructured manner. So there was not the same amount of sampling effort across elevations. If I remember correctly, richness is higher at the foothills. Yes, it is. But I need to do more structured surveys in order to be absolutely sure of that, and also combine both trapping and acoustic recordings. Back then, I wasn't doing recordings because the point of the survey was to make an acoustic call library, which would allow us to identify bats later on.
Excellent, thank you. Okay, more questions. So Jorge Noriega says, hi Rohit, nice talk, congratulations. I was wondering if the patterns of bats could be correlated with the patterns of their potential preys.
That's a good question. We haven't looked at it as yet. It's hard to answer. We do have I know somebody who has data on moths from similar elevational gradients, and I think it's great to compare these two. And thanks for that suggestion. I really don't know. We haven't looked at it, but I suspect the patterns may not just hold true across the prey. Across the prey species, but also might hold true for various other taxa. For example, you see similar patterns with plants as well in the Himalayas.
Excellent. So this question is done. And then we have another one by Bhageratan Murugabel, and says, excellent talk, Rohit. Are you planning for having more replicas in the 3,100 meters elevation? Also, what do you expect would happen to the patterns if you include that site into your analysis?
Yeah. Okay. So offhand, I can say that yes, the patterns would be pretty similar to what we've observed at 3,000 meters. The species that we've detected at 3,000 and 3,500 do not change, are pretty much the same. So the patterns would be I would expect the patterns to be just the same, even if we added more spatial replicates.
Another question.
Oh, okay. No, I think it's just.
Well, thanks. Congrats on the paper, by the way. Yes. Brilliant. Thank you. Thank you all for participating. We have another question in. So, Trevor Price, what about the east, which has a good gradient?
Yeah, I would be most interested in going eastwards for this, and yeah, looking at Trevor's papers comparing east and west diversities in birds. I'm even more motivated to check these patterns. I really can't tell what I would expect. I would probably expect a mid-elevation peak in species richness in the east. And but what implications it would have in functional and phylogenetic diversity, it's anybody's guess.
Fantastic. Okay, thank you. So there's another question from Diogo Ferreira. Says, great talk. And he follows up saying, if I understood correctly, for the analysis, you only used acoustic data. So all the species captured with mist nets were also recorded. And why only using acoustic data? Few captures?
Yes. Yeah. Thanks for the question, Diogo. Yes. So the reason for using the acoustic data was few captures, and most of the species in the acoustic data, I mean, that were caught in the mist nets were also recorded, but some of those we can't tell apart very easily. So it does underrepresent, like I said. But yeah, and then using acoustic data also had some small problems. For example, and I mean, yeah, okay, the problems are twofold. So we caught one species of fruit bat just once at the higher elevations, but we had to exclude that from the data set because they don't echolocate, so we did not detect them. But on the other hand, there was the free-tailed bat, the Tadarida, which Diogo as a bat biologist would know. So the Tadarida flies very, very high, and as you know from Portugal, and we couldn't catch it, but we recorded it very often. So we had detected it, but we didn't have trait data because we hadn't caught it back then. Yeah. So the point is that yeah, and the reason why we did not combine the acoustic data and the mistnet and the trapping data is that they tell two different things. They tell two completely different things. It's really incorrect to put both of them together as a measure of relative abundance, because on the one hand you have actual captures, on the other hand you have detections which could be of the same individual again and again. So it's a bit tricky to combine these two to obtain a measure of relative abundance.
Excellent. Okay. So now we are done. No more questions for now. And I think that Karen is about time to move on.
Yeah, I think so. Our next speaker, Nina Witteveen, who also won an honorable mention for talk, she wasn't able to join us today. But we have her video recording from the conference. And so at this point, I just wanted to say a huge thank you and congratulations to both Oskar and Rohit and Nina for the awards. It's been really nice to see your talks because I didn't catch them at the conference just with all that was happening. So it's really great. So thank you for joining us and congratulations. Sandra, do you want to say anything before I go on? Because I assume that not everybody will remain for the video. And so.
Basically, thank you so much for the speakers, for the great talks. And then let me introduce just briefly Nina.
Oh, yes. Thank you.
So Nina as well was the third winner. And she's currently a PhD student at the University of Amsterdam. And she's very interested in assessing the legacies of past human activities, mainly in Amazonia. And as she mentions, she says that she's very eager to contribute towards a sustainable future.
Hi everyone, welcome. My name is Nina Witteveen, and during this presentation, I would like to take you to the Andes, to the mid-elevation, where I think the view is pretty nice. The research I will be talking about during this presentation was recently published there, and it was a part of my master thesis at the University of Amsterdam, supervised by Crystal McMichael and Will Gosling. So in the Andes, as the elevation changes, so do the environmental conditions and the vegetation types. With all the way in the highlands, you have the biomo grassland, and in the lowlands, the Amazon rainforest. And in the last 30,000 years, when we moved from our ice age to our current interglacial, globally it got warmer. And 3 species migrated in response to that. In the last 11,000 years, during the Holocene, in the tropical Andes, temperature was more or less stable, but precipitation fluctuated a lot. From fossils from manioc, also known as yucca or cassava. And then in Ecuador, the first evidence of maize agriculture is from 8,000 years ago in the highland and 6,000 years in the lowland. So both in the highlands and the lowlands, people were there and they settled there for quite a long time ago. But we know relatively little about what happened in between at these mid-elevations. And because the environment changes so rapidly as you move up and down that mountain slope, the mid-elevations is actually a hotspot of biodiversity. So the aim of our research was to find out how the vegetation changed exactly at those mid-elevations, going back 30,000 years. So this is Campo Libre, over here in the northern Andes in Ecuador, at 1,800 meters above sea level. And this site is an old floodplain. So it's very swampy, it flooded often, and over time, as it got disconnected from the river, it got drier. It's surrounded by tropical forest vegetation. So right here at this site at Campo Libre, my colleagues, they collected a very long soil core of over 4 meters. And for each centimeter of soil throughout that core, we analyzed both the soil and several proxies in that soil. So next, we constructed an age depth model. So for each depth, we found out what the age was to look not only at the change over depth, but actually at the change through time. So what about those proxies? So in the soil of this swampy forest, pollen degrade, but phytoliths preserve. So phytoliths are super small plant fossils from silica. And I studied those under the microscope to look at the vegetation history. Charcoal is also assessed under the microscope to look at the fire history. Loss on ignition and particle size of the soil was analyzed to reconstruct the past river dynamics. And a DCA and CONUS analysis was used to look at the past vegetation, using all the different phytolith fossils that are identified in each centimeter of soil. So I would like to tell you about the vegetation change through time, using the phytolith diagram results over here, and an impression of what the vegetation in the past would have looked like over here. So let me first walk you through these figures. So over here you can see the different tree species that are shown in this panel. And the phytolith diagram on the y-axis is time, and the top is modern, and the bottom is 30,000 years ago. So this box over here shows the total phytolith fossils, the total palms, total trees and total grasses, and it's shown in influx, so the amount of phytoliths that were deposited in the soil yearly. Then over here in this box, 3 of those grasses are shown, and one of them is the maize crop. And then this box shows the first DCA axis. So a DCA is used to look at the past vegetation change and also how big the vegetation change was. So spoiler alert, there was a large vegetation change over here at this peak. And then the zones CL1, CL2 and CL3 are the significant vegetation zones indicated by the CONUS analysis. So now that I've walked you through this figure, let's move on to the results. So during the late glacial period, in zone CL1 from 30 to 18,000 years ago, Campo Libre was a floodplain consisting of several grasses, some wet adapted grasses. And because it was a part of the floodplain, phytoliths from the regional vegetation also washed in. And then still in zone CL1, but after the last glacial maximum, Campo Libre is no longer a part of the floodplain. So we lose some of that regional vegetation. And you can see that over here when this grass disappears. And also as it gets warmer, palms first enter the landscape. You can see this peak over here. And specifically, we found fossils from Euterpe and Geonoma palms. Then in zone CL2, 13,000 years ago, when it got drier, we see the largest vegetation change because the swamp got turned into a palm forest. So again, we still find these fossils from Euterpe and Geonoma, but now we also find a lot of fossils from Bethynia, Dictyocarium, and Bactris palms. And because it got drier, some of those wet adapted grasses disappear, such as this one. Then in zone CL3, as precipitation increases again, we can see that some of that swampy vegetation actually grows back. So you see that the trees increase again, and also this wet adapted grass comes back. And now the site is really a mixture of trees, palms, and grasses. Up until 4,600 years ago, still in zone CL3. And that's when we see the first signs of people. And you can see that right here at the maize fossils. So people enter the landscape to grow some maize crops. So fire was actually completely absent from this site right until 3,300 years ago, when we see the first signs of regional fires. So large pieces of charcoal indicating local fires were not found. So this system has evolved and adapted completely in the absence of fire. So during this time period, you still see a peak of palms, and this was actually mostly fossils from Bactris palms. So perhaps the abundance of Bactris during this period was influenced by this past human activity. So how do we know that the vegetation changed because precipitation decreased and then it increased again? Well, that's because we took the results from the phytolith diagram here and we compared it to global and regional climate records from the literature. And the largest vegetation change in zone CL2 and indicated by this DCA peak here, it happened exactly when regionally it got drier and then it got wetter again, as you can see here. So because this climate and vegetation records, they correlate so well, we think that the vegetation changed in response to these climatic changes. So my take-home message is that these ecosystems at the mid-elevation in the Andes, they were very dynamic and very responsive to past climatic changes. Because the biggest vegetation change occurred not when people entered the landscape, but actually when there was a climate change. So therefore, I expect that this biodiversity hotspot is also sensitive to our current climate change. So I would like to thank all the other co-authors and all the people that worked with me on this project or made the research possible. It was a very fun project. And I would like to thank you all for listening. So I moved to the lowlands. I dedicate my PhD to vegetation change in Amazonia. So please check out my Twitter or the blog of Will Gosling if you're curious. Feel free to throw all your questions at me now. Looking forward to discussing it. Thank you.